In this presentation, we are going to look at osmosis in living tissues, evaluating the effects of various solutions on an epidermis peeled from the lower epidermis of tradescantia. My name is Lenya Jude Fanba. I am a holder of a Bachelor of Education degree in Life and Earth Sciences from the University of Marua, Cameroon, and a Doctor of Philosophy degree in Clinical Biochemistry from the University of Chang, Cameroon. This experiment is based on osmosis in living tissue. As a reminder, osmosis is the movement of water across the cell membrane from a solution of high water potential to a solution of low water potential. The experiment is also based on the mechanism of opening and closing of stomata. When guard cells absorb water by osmosis and become turgid, the stoma opens. On the other hand, when guard cells lose water, they become plasmolyzed, collapse, and close the stoma. The vacuoles of tradescantia epidermal cells contain a pigment called anthocyanin, which makes osmosis visible through color changes. To better understand this experiment, let us look at the structure of stomata. A stoma is made up of two semicircular guard cells surrounding a pore. The guard cells are specialized epidermal cells containing chloroplasts and regulate the opening and closing of stomata. Stomatal pore opens into the intercellular spaces of leaf mesophyll. As hypothesis, we can say osmosis occurs only in living tissues and not in dead tissues or non-living matter. We are going to use a set of materials and solutions, including a tradescantia leaf, which is the biological material, the living tissue from which we will obtain an epidermis. We need microscope slides and slide covers on which to mount the epidermis for observation. We also need a dropper to apply solutions on the placed on the slides. A light microscope will be used to observe the stomata on the epidermis. 3 beakers will be used to hold the various solutions, and a wall clock with a sweeping second hand for timing. We are going to use 3 different solutions. Distilled water will serve as the hypotonic solution. A 1 molar sodium chloride solute by dissolving the molar mass, 58.44 grams of the salt in 1 liter of distilled water will serve as a hypotonic solution. A 1 molar sodium hydroxide solution made by dissolving 40 grams of pellets in 1 liter of distilled water will serve as a corrosive solution, which will destroy the epidermis and provide the control. From a fresh leaf of tradescantia, we will obtain the lower epidermis by the break and pull method as follows. Slightly fold and break the leaf near the edge. This creates a natural break in the epidermal layer. With the hand, gently pull the broken edge against the leaf to peel the transparent epidermal layer away from the leaf surface. Make 3 of such peelings and place each on a clean and labeled microscope slide. Apply a drop of the solution on the slide. Cover with a slide cover and quickly observe under the microscope at times 40 or times 100 magnification. Then allow the slides for 10 minutes and then observe again. A sample report of this procedure will be: a fresh leaf of tradescantia was broken from the upper surface near the edge. The short edge was pulled against the leaf to peel off the lower epidermis. 3 pieces of the epidermis were placed on 3 different slides labeled distilled water, 1 molar sodium, and 1 molar sodium hydroxide. A drop of the respective solutions was placed on the 3 pieces of epidermis using a dropper. The epidermis on the slides were each covered with a slide cover. The prepared slides were observed under the microscope at times 100 magnification, and the openings of were noted. The slides were allowed to stand for 10 minutes. After the 10 minutes, the slides were again observed under the microscope at times 100 magnification. The openings of stomata were again noted. Observation of the slides showed that in distilled water, the guard cells took a crescent shape or shape of the half moon, causing the stomata to open widely. In 1 molar sodium chloride solution, the guard cells collapsed and closed the stomata. In 1 molar sodium hydroxide, the cell membranes were destroyed and the chloroplasts dissolved in the guard cells. As interpretation of the results, we have the following. For the epidermis in distilled water, we can say distilled water is hypotonic to the epidermal cells. That is, it has a higher water potential. Water thus moved into the cells by osmosis. The The cells became distended under the influx of water. The expansion of the outer and inner walls of guard cells caused them to curve, and the curved guard cells caused the stomata to open widely. For the epidermis in 1 molar sodium chloride solution, this solution is hypotonic to the epidermal cells. That is, it has a low water potential. Water moved out of the cells by osmosis into the solutions. The epidermal cells became plasmalized and shrank. As for the guard cells, they collapsed, and the uneven retraction of outer and inner walls led to closure of stomata. For the 1 molar sodium hydroxide solution, the solution is corrosive. It thus killed the cells, destroying the cell membrane and dissolving the chloroplasts of guard cells. The cells lost their selective permeability due to denaturation of the cell membrane and its transporters, thus no osmosis occurred. This setup served as the control as the cells were killed, and no evidence of osmosis was observed. From these results, we can conclude with affirmation of our hypothesis that osmosis occurs only in living tissues and not in dead tissues. To arrive at these results, we took some precautions that helped us to have reliable results and also for safety. The dishes were appropriately labeled with names of the solution to avoid confusion of the solutions. As a reminder, during examinations, solutions will be coded with letters, say, A, B, or C, and you will be using only the terms hypotonic solution and not distilled water, hypotonic solution and not sodium chloride solution, and corrosive solution, not sodium hydroxide solution. We have come to the end of this presentation. We are through with osmosis, and our next presentation will introduce us to food tests. Thanks for your time. Hope you enjoyed following up the presentation. Please subscribe to this channel, like, and share. By subscribing, you will be getting notifications when new presentations are available.